A good morning or afternoon to you. Thank you for joining us. I'm Jan Reichert, Executive Director of the Antibody Society. Today's webinar is one in a series designed to inform and educate our members, as well as the broader scientific community, about topics relating to antibody discovery and development. Our expert speakers, Ben Osborne, Jake Glanville, and Joel Christian Andrade, will discuss best practices relating to biolayer interferometry and its application in screening of a broadly neutralizing antibody of snake venoms. Please note the webinar is being recorded. The Gator Bio presentation can be downloaded from the Materials tab in the viewer. Please do add any and all questions to the Q&A box in the viewer, and those questions will be answered at the end of the webinar. Without further ado, I now turn the show over to our first speaker.
Thank you very much. Thank you, Jan, for that introduction. My name is Ben Osborne. I am a field application scientist with Gator Bio, and today I'm going to briefly cover some topics related to biolayer interferometry, what it is, how to do experiments right, and then I'm going to hand off this presentation to two guys who know how to do this right. And they're doing some very exciting and awesome work. So I'm very eager for all of you to see that presentation. Briefly, I want to introduce what and who Gator Bio is. Gator Bio is a company that specializes in biolayer interferometry equipment. Biolayer interferometry is a technology that's been around since 2001, where Octet, the Octet instrument was first developed in 2001, founded by Hong Tan. And as over time, the story of the company, it was purchased and handed off to several different companies. And recently, Hong decided that he wanted to step back into the BLI sphere, and he founded Gator Bio. And we now have two systems, the Gator Prime, the Gator Plus, and they are next generation systems for interferometry applications. Gator Bio is a company with almost 100 employees. We make all our probes in the United States. We don't have supply chain issues for US customers. And we have a number of exciting innovations taking biolayer interferometry to its next level. Things like reusable streptavidin probes, AAV-specific probes, amplification procedures and technologies for measuring really small amounts of something that you're trying to quantify. And we have over 200 instruments around the world in places in the EU, in Israel, in Korea, as well as many places here in the United States. Now, let me explain a little bit about what biolayer interferometry is, because I'm sure that's a question you probably have. Biolayer interferometry is, if you go back to your high school physics classes, you might remember that if you have waves that are in line with each other, that are in phase with each other, that they amplify and create larger waves. Whereas if you have waves that are exactly out of phase with each other, they interfere deconstructively, they cancel each other out. Biolayer interferometry takes advantage of this principle in a very simple way. So you might imagine that we have a light at the end of our glass probes, and all the light bounces back at the end of that glass. And so all the light is in phase. But as biomolecules bind on the end of the sensor probe, it creates a new protein layer on the surface, and that creates another phase transition where light can reflect back that's a few nanometers further down. This causes some light to reflect at the glass and some that reflects at the end of this protein coating layer, and that ends up having two light waves that are out of phase with each other. We can measure that decrease in light intensity because of that interference and use that to generate real-time measurements of biomolecules binding on the end of a sensor probe. And this has a number of applications. So as I've talked about earlier, you can use it to quantify antibodies that you're manufacturing. You can use it to quantify AAV capsids. How many intact capsids do you have inside your sample? You can also do things like real-time kinetics measurements, where you're observing real-time on and off rates. In this example, we're doing protein A to human IgG, but you can use this for a variety of other applications for whatever you're trying to develop. And you're going to see a lot of kinetics data coming up. You can also use it for epitope binning, where you bind an antibody to an antigen, and then you see if a second antibody can bind that antigen while the first antibody is bound. That allows you to discover, do they share the same binding site or not? Now, one of the advantages of the Gator system is that it's a very simple label-free analysis. You can we offer a variety of sensor probes, and I'll show you those here in a minute, that allow you to bind a wide variety of biomolecules on the surface of your sensor. And this is compatible with purified or crude samples, and you're able to recover the sample afterward because the probes only take a small amount out of your solution. The Gator system is also relatively low cost and has very little maintenance required. And by that I mean, many of our sensor probes are developed in such a way that they allow you to regenerate them multiple times. You can use a protein A probe or our brand new generation 2 human FC probes. You can use them a dozen times or more, repeatedly using them and recycling them for multiple experiments with very little loss of signal intensity. We also offer a cutting-edge remote lamp adjustment feature. It's a driver in our software. We found that with a lot of companies that are out there, that the lamp intensity can go down over time. You have to send a technician out, replace the lamp. But we found that if you're starting to have some interesting odd data, that we can adjust the lamp remotely and extend the lifetime of your instrument and get you back on your feet sometimes within 24 hours. We offer a 2-year warranty standard with all our instruments because they're very reliable, and we have very reasonable service contracts. But the most important things that you need to understand about BLI is that it's not a specialist instrument, that the software is intuitive, that it's very fast to get your samples through. We read 8 samples at a time, and we have a team of field application scientists who will get back to you if you have any questions about your data. You send your data to a field application scientist, they get back to you within a business day. That's our model of customer service. That's what we try to do. And it enables people to really do the science that they want to do very quickly. So right now I want to take a brief moment for this is sort of a something for everyone presentation. I want to present some of the basics of biolayer interferometry, as well as some good practices as far as kinetics goes. A lot of people who are watching this presentation might have a BLI instrument and might want to be able to do the kinds of experiments that you're going to see here later. And I just wanted to talk about some of the tricks that I've found to improve the kind of data that you can generate. First thing that you got to know when you start BLI is which probe should I use? Now, if you're doing a niche application like AAV quantitation, we have very specific probes for that. Our AAV-X, our AAV9, and our high sensitivity AAV kits all accomplish that task quite well. But we have a variety of probes for antibody screening and antibody screening purposes and developing assays involving antibodies. That includes our protein A, protein G, and protein L probes. We have anti-human FC probes. We have one that's really more specialized for kinetics and repeatability now, with very high regeneration capabilities as well. And we have a fantastic anti-mouse FC as well if you're doing mouse antibodies. But we also offer some of the other standards like anti-His and nickel NTA. So if you want to, if you've produced His tagged proteins, you are able to throw them on the instrument right away. I also want to talk briefly about the power of our SMAP probes, which are streptavidin probes with extremely high sensitivity. You'll see some data later on where I'm working with a customer that's using molecules as small as 300 Daltons, even down to 150 Daltons, and we're able to see the kinetics and the binding signals for those molecules. And we also offer, as I mentioned earlier, streptavidin probes where you can cleave off the streptavidin and completely replace it. Choosing the right probe for your experiments is key. If you're doing epitope binning type experiments, our generation 2 human IgG-FC probe or our protein A probes may be the right fit. It just depends on how specific you want it to be, how good the kinetics you need to look. But we offer a variety of probes and even offer custom probes depending on the application you want to accomplish. The next thing you need to know is whenever you do an experiment with BLI, you have to know what buffers you're using. Typically, PBS is a great start, but probes are compatible with a variety of buffering agents, MOPS, heaps, whatever. It probably will work with the probes. You can also get away with addition of surfactants or BSA. Surfactants can often be in there up to 3%, and you'll be just fine. Typically, we do tween at around 0.2%. Our Gator K and Q buffers have a small amount of BSA and detergent, tween 20, to enable reduction of non-specific binding in your samples. But see, one other thing you've got to understand is that when you acquire a protein sample, let's say it's produced by another department in your company, or you purchase it commercially, you don't always know what's in your buffers. And I see this all the time as a field application scientist. Glycerol doesn't come out when you do lyophilization. And so you'll resuspend your protein and you'll find that there's glycerol contamination. Here on the right is a small amount of glycerol contamination. You see these jumps in the signal. That's indicative of that. So if you didn't manufacture the protein yourself, you have to pay attention to the buffer conditions or additives that may be present. Something else that you need to understand is that it's actually quite simple to be able to adapt whatever probe you're working with to the assay you need to do. So, for example, if you're using a streptavidin probe, you should always assume that commercially produced proteins are AVI tagged, and you can simply block, after you load your probe with what you need to load it with, you can add a biocytin dipping step. You dip the probe into biocytin or something else, and that will completely prevent any further binding non-specifically to the probe. But we offer a variety of other, but there are a variety of other ways that you can block things. Like if you're doing epitope binning and you have an anti-FC probe, you can put in a isotype control you get from in vitrogen, and that will completely cover up the remaining binding sites on the probe so that your epitope binding results are clear. But to prevent non-specific binding, we generally recommend detergents, adding some BSA, or playing with your salt concentrations can often help. There's also a number of useful software features that we offer here, and that is that you can play around with your analysis features, save that analysis, and then go back to it and create a new analysis separate, and then allow you to toggle between those and see what the difference is as you're playing around with things. And that's one recommendation that I make for people as they're getting started with the software is just click buttons. You're not going to break anything. The software is very intuitive, and it doesn't allow you to screw up in a lot of ways. Also, a handy tip: you can screen multiple kinetics assays in line with each other all at the same time. All you need to do is sort each of these curves by color. So in this case, I have here a 750 Dalton molecule, a small molecule binding onto the surface of our probes. These yellow curves are a 350, and these green ones are a 320, I believe, Dalton molecules. You can see the binding curves very clearly here on our high sensitivity probe. They're separated by molecule here, by color. And when they're separated by color, the software automatically splits them and analyzes them globally for you. It's a very simple and intuitive system. Makes it very easy to analyze your kinetics. Now, stepping into kinetics analysis, some things that you should be thinking about when you design a kinetics assay is that you need to understand what is the ideal loading concentration to avoid avidity, blocking, or mass transport effects in your later data. We recommend people to load their probes to 60, 70% loaded. All you have to do before you run a kinetics experiment is simply load your probe with a titration of your ligand that you want to bind to the probe. And this will allow you to very quickly identify what is the ideal concentration that you want loaded on the surface of your probe. I also recommend whenever someone starts doing kinetics that you run a particular set of controls. The first control is you have a probe that's loaded just like all the rest, but then you have zero analyte in the solution. That makes sure that you don't have any buffer effects that may be affecting your data. The second control is I always like to leave channel 8 where I don't load the probe with any protein, but then I do load it with analyte, with a high amount of analyte. And that allows me to detect when I have a non-specific binding issue. For example, in this case, I had someone had an antibody that they had purchased commercially, and they were using a streptavidin probe. They had streptavidin bound their ligand, and they were looking to see how well the antibody bound to that ligand, and the antibody was biotinylated. And this allows us to very quickly identify what the problem was and move forward with better assay development and design and blocking. Some other tips for you is that no matter what your experiment is, 95% of the time, it's best to assume one-to-one binding. Even if you're playing with antibodies which have 2 binding sites, those bindings are independent and individual. So you can typically assume one-to-one binding. However, sometimes you see binding curves that look like this, where you have continued binding like this. And those are caused by a handful of things. Let me give you a couple of tips. If you're seeing this in your own BLI data, here's some things that you can try. And the first is that your analyte concentration may be just a hair too high. Using a lower, you can see that we're getting up to 5.5 nanometers shift. I would recommend that you lower the concentrations that you're using here and try to get a standard curves that look correct. You'll see in just a minute what that looks like. This also could be avidity, that your probes were overloaded in an earlier step. So watch your loading. Make sure, you load it at 60%. Another cause of this could be some nonspecific binding due to other proteins that may be in the solution. Again, you've got to know your buffers when you're trying to diagnose what's going on. So to deal with biphasic binding, you typically will reduce the concentration of analyte. You can invert the order that you bind things to the probe, and that will often help as well. I've also found that adding a small amount of detergent or increasing salt concentrations can be very helpful. But here are a couple of other suggestions for you if you find yourself having these difficulties. Again, I want to point out, I'm talking about here's all these ways to fix if your data doesn't look particularly good. But note that these fixes are actually quite simple, that developing an assay, once you've got it developed, it's very repeatable. They're able to get the results that you're looking for consistently. And again, you'll see an example of that here in a few minutes. So I am often asked, when can I trust the kinetics data that I'm receiving? And this, I think, is the most important part that I can talk about. And that is, the first question you need to ask yourself is, does the curve fit approximate the trend of the data well? And that goes back to, in our software, it's how you're aligning the data between the steps, what kind of fitting models you're using, how long did the experiment run, some of those things. Again, we have a rapid response FAS staff at Gator Bio. So if you are having troubles with any data that you're receiving on the Gator, you can send us the raw data files and we'll open them up. We'll have a Zoom meeting with you and we'll walk you through the steps to design this assay better or work with what you've got. Often we can salvage the data you have pretty well. Is my R squared greater than 0.95? How good is the curve fit? This is an objective way of measuring, whereas this is visual representation. This is an objective way of saying that the fit is representing well and that the kinetics data is good. And finally, looking at the residual graph, are you within 10% of the largest signal? Because if you're not, then you aren't representing your data very well. But like I was talking about earlier, it's very important to try, once you get sort of a preliminary idea of what the KD is, you want to do a assay that is within 10x of that ideal KD concentration. That will give you curves that look like this, where it usually flattens out to equilibrium on the highest curve and goes on down. This is an example of what it looks like when it's too high. Here's an example of what it looks like when you're running something that's too low. This is to serve as a reference. These slides are posted online, so if you are struggling with any Gator or any BLI data at all, this is something to be looking for. Finally, you want to ask yourself, is your K on and your K off error less than 10%? So here's where the error is. Here's what it looks like inside the Gator software. Here's where the error is posted, as well as your K off rate. And this is what this. And then finally, does your curve fitting data, which is what shows here on the bottom spreadsheet on the Gator software, and your steady state approximations, which is what's shown here, do those give you the same result? And I'd be happy to go into more detail about this. Feel free to send an email to support@gatorbio.com if you have any questions about what I'm talking about. But if they're giving the same result, you can be confident that the data that you're receiving is in line with itself, that it is working well. Again, this is an example of small molecule data. This is a 750 Dalton molecule. And you can see very clearly that we have very good and clear binding kinetics, and that the data is very in line with itself. And now, real quick, I want to talk about 1-point antibody screening tips. Oftentimes, you'll find yourself where you're screening 100 different antibodies to a target. And the way that you can identify what is and what is not a good target is that you're looking at, what is my response? How high does it actually get in my association phase? And how slow is my off rates? And the off rate is listed here as the K off. It's very important that you pay attention to these little parameters. It's very easy to get fooled when you look at just one response. When you have just this graph on the view, it looks very big. Pay attention to the way it's binding. Pay attention to non-specific binding effects. Pay attention to its off rate. Those are all the keys to identifying good candidates for the screening that you want to do. And now I'm going to hand it off to my friends Jake and Joel. They have done some awesome work in using these exact techniques, and I'm very excited for you guys to hear what they're going to present.
All right. Well, thanks again for that. Yeah. So, and thanks for having us on today. So, hello, everyone. I'm Jake, the founder and CEO of Centivax, and with me, Joel.
Yeah, I'm Joel. I'm a scientist at Centivax, jack of all trades, master of none. So, go ahead, start.
So we're going to share with you today this project that has been near and dear to my heart for about 5 years now. Some of you may know me from my previous company, Distributed Bio. Since then, I've created Centivax. What we focus on are broadly neutralizing antibodies, either eliciting them in vivo, spectrum vaccines, or in work you're going to see specifically as a cocktail of BNAbs for the treatment of antivenom. This particular project involved us taking the instrument and generating, in addition to analyzing a number of samples, we generated a new assay that was particularly important for the analysis of a broad spectrum antivenom for a snake. And so we'd like, okay. I might start just saying like why, if you live in the United States, unless you live in like Arizona or parts of California, Rocky Mountains, Florida, you may live in a place where you say, why do you care about snakes? And so I want to tell you why. So first off, I just mentioned there actually are a lot of states where snake bites are happening, but it's a much bigger problem than you may realize. Around the world, there's somewhere between, 2.7 envenomings of over 5 billion bites. We don't even know because a lot of the people who are being bit are out in rural areas where they may not be reporting that back in a way we can gather the information. What we know is that there's 300 to 400 that are catastrophically injured in a non-recoverable way. They lose a limb, their leg is amputated, disfigured, and then there's 100. Estimate 100 to 125,000 people that die every year from snake bite. So it's a really big problem. It's one of these problems that happens mostly somewhere else. The Health Organization recognizes snake bite is one of the major neglected tropical diseases. It's one of these areas where there is actually one of the oldest antibody-based therapeutics, and it's developed. There's improved that much over time. It works. There are a number of limitations, and through those with you now. So, Paul, you want to take it?
Yep. So what really compounds the problem, and I think maybe to a layman's eye would be really difficult to understand, is that there's over 3,000 different species of snake. Not all of them are venomous. I think certain estimates put it at over 600 different venomous species. And about 200 of them are capable of killing humans. So that's pretty tough if you think about it from that point of view. You have a diverse set of snakes from different geographical regions. And I'm here to continue to compound the problem. But to simplify it first, there are two main families, the Elapidae and Viperidae, or elapids and vipers, where most of the venomous snakes come from. And the way that venomous or potency is used is kind of interchangeable at times, because in many cases, as you'll see with the examples I have on this particular slide, snake venoms can be both incredibly lethal within 20 minutes to an hour, or at sometimes causing death in 3 to 4 days, but severe local damage leading to death.
And the challenge with the diverse, the 550 to 600 venomous snakes currently made is that for a single species or a species that are very related to each other. That has created the market of antivenom, pretty predominant market that is people in developing countries that have a lot of resources, and you've just made it harder to generate a working profitable business to make antivenom for them because you're only serving one little sliver of that market. An antivenom for cobra, antivenom for some taipans, so forth, all around the world. And that also means that many of markets are neglected, that there are not venoms for many of these snakes. It's also tough for people who have been bit in an area where there might be many different types of snakes. Because what happens is if you get that person to a medical Going to say, "Oh, did you get bit by a snake? Oh, did you do me a solid and like go back out and like look around the bushes and tell me what the species that even exists in antivenom, either in my facility or in the world?" Great. And that's part of the reason we're focusing on this, to see if there could be a single product serve and unify the market and to treat without having to do that testing or estimating of whether the right antivenom exists for the bit with.
Sure. And like, for example, the two on the previous slide here, so we have like the black mamba and the green mamba. Those are primarily found in sub-Saharan Africa. Those could kill you in 20 minutes, but you'll often find them deep into the jungles where they're also incredibly aggressive. So while you may get a single dose of that venom from one bite, these are likely to bite you 3 or 4 times, and you are probably 30 minutes to an hour out from any close medical facility, like Jake would note. Even more complex, for example, the monocled cobra here, primarily within Vietnam, Thailand, and Malaysia. Within those 3 countries, there's severe diversity where the LD50, which is the mean time to killing for about 50% of individuals, drastically changes. So if you were to be bit with the monocled cobra in Thailand, you are likely to have a lethal dose almost immediately. Whereas if you were in Vietnam or Malaysia, you'd have a completely different composition within that venom. So you can see here how that changes. So moving to this next slide.
And one extra point on that, the genetic variation, population genetic variation, even snake species, like what was just mentioning, also means antivenoms for that species don't work equally well across the range for that. So it's a mess, I guess, is the point we're trying to say. And it gets even worse, as Joel's about to describe, with respect to the content of the venom.
Sure. So 2 different mechanisms, primarily within these 2 families, work with for vipers, causing a lot of local damage. This is really done through PLA2, SVMP, and SVSP. Those are proteases and proteinases, which cause local death and eventually prevent, in most cases, say for the snake's natural prey, which would be some type of rodent, severely slowing their movements, making them easier to digest, and eventually causing death. These are the vipers where you would not consider them as quickly lethal compared to the elapids, but they will cause death if it's untreated or lead to severe disabilities such as amputations. For elapids, the main toxin here are 3-finger toxins, and these work directly on creating paralysis within the victim. In our case, the one we'll talk about later is the long neurotoxin, which binds very strongly to the nicotinic acetylcholine receptor, which can cause paralysis in the lungs and eventually death almost within 20 minutes at the earliest that we've seen. Not only that, there's severe heterogeneity within all of the different 3-finger toxins. So long neurotoxin might be one component, but there's also short neurotoxins and cytotoxins. And between individual species, so say we're talking about the black mamba, there can be variation between individuals within it. I noted earlier, geographically, it can change drastically in the composition of the venom. And even within, we were talking to some researchers the other day where they were looking at what is known as a clutch, or what you would think of like a litter for pets. So a bunch of baby snakes. Within a small group of those infant snakes, there was some differences in the expression of different venoms there. So the goal here, at least going forward, would be to find something that not only broadly neutralizes the different components in them, but also can address a conserved site among all of them.
So let me set the stage a bit. So we've just told you that there's 550 to 600 venomous snakes around the world. We've also just, each of those snake venoms could have 5 to 70 fractions or different proteins in that venom. You can see why there is no universal antivenom, and why a reasonable person would say, well, that's impossible. This is why we're stuck in this world where we have specific polyclonal antivenom. Usually they take venom and they inject it in horses or sheep, and then they take their sera, they cleave off the IgG FC to create Fab or Fab2, and that's the product. That's been the way it's been for 125 years. Small improvements. What we're going to share with you today is we think there actually is a mechanism to address this, and it comes in two parts. One is that, yes, while there are 550 to 600 venomous snakes, there's really about 20 that are responsible for the vast majority of and mortality around the world. And there's a whole bunch of them that, yeah, they're venomous, but if you get bit by them, it's going to be annoying, rash, you might get some swelling. But to create a universal, you really just need to make sure you cover the ones that would actually cause damage. So then if the snake was going to kill you or make you lose your leg, and if it wasn't going, and taking the antivenom was harmless and you're either. With respect to the toxins, laziness. So even though there are a huge number of snakes, they have many different fractions, and there's polymorphism in the populations, also about 10 families, 10 homologous families of toxic types. Look at those pie charts, and then beyond those pie charts, what we know about the toxins, not all of them are equally important. We estimate there are 5 toxins that are the thing cause you death or cause, and that's the phospholipase, PLA2, the SVs, the bases, SVMPs, serine metalloproteinase, and the 3-finger toxins, of which Joel mentioned there's a couple subclasses. Then you can walk toxins. So a universal and a broad spectrum venom act, provided that you can identify broadly neutral to knock out each things. Like would serve on the planet by that market and would enable a number of other advantages by virtue of the antibody, avoiding a whole sickness problems that are inherent to administering non-human and anti-sera. So that we're going to be sharing the question of to what extent do broad neutralizing antibodies against antivenom exist. So of those toxins, to long neurotoxins. The long neurotoxins are a 3-finger toxin. They are the most neurotoxin that exists across them. Snakes that are called elapids. Those are your neurotoxic snakes. The reason we thought a good place to start is as follows. On the left there, showing you the nicotine receptor, target of the long, and we've highlighted where the binds. The long neurotoxin shown on the right region of its epitope. The nicotinic acetylcholine receptor is super conserved. That's like that heat. Any purple revolutionary. You notice that this thing looks nearly identical all the way out to fish. And that's pretty cool for the snake because it means that its venom to be effectively, a mouse or a lizard, gecko, a fish, and that's super useful. It's applicable across many different types of food sources. But that imposes a constraint across all of those venoms. And that is that they are not able to change part of the base their toxins, because if they change, they can no longer engage the single across life that they target. And that creates an Achilles heel at that surface where I said, let's start here, because I think because they have that ultra conserved face, if we can identify a broadly neutralizing antibody against it, could neutralize long toxin from across the venomous snakes. And so that was the exercise. We had a little bit of a trick up our sleeve. We were looking for humanized antibodies or use, discovery libraries, of course. But I was looking around to try to see someone who was a clumsy snake researcher who had been bit a couple times and therefore might have human antibodies against snake venom. And during the search, 2017, found this guy named Tim Free. This was a person over a 17-year and 9-month himself 706 times, carefully escalated doses of venom from 16 different species, across at Lapidae and Vipera. I'm showing all the species down there at the bottom. And he had taken these like meticulous notes where he'd recorded the micrograms, milligrams of escalated wet weight of the species, injectometer of swelling that occurred afterwards, self-reported discomfort score, many of which I'm illustrating on the right for a small piece of loading schedule that he did over 2 years for the Dendroaspis. You can see as he increased the dose, there was this precipitous drop suddenly in the swelling, find the appearance of immunity, and then also a reduction in the discomfort score. So I'm sure my initial reaction was, I'm sure, was the same as yours. I was just like, oh man, this is what is going on. But I felt when I looked at it that because of that so many people are dying every year and being injured, and there's either nothing for them or what they have are these polyclonal horse products. I don't know, to take a look further at this guy's repeated effort for nearly 18 years of self-immunizing, that he might have applied selection pressure for breadth. He might be an ideal place to look for fully human, broadly neutralizing antibodies against shared sites on snakes. So we obtained in a interventional study 40 milliliter of blood, did it into the serum and into the B cell. From the serum, we ran a study to characterize whether or not he had that history resulted in an enhanced serum response. And from the B cells, we generated a library and characterized whether there were signatures of repeated affinity maturation history of the B cells that was his reported. The same thing, I'll take the somatic hypermutation thing. You're muted.
So in this case, we saw that there was a strong amount of serum response when it comes to ELISA when taking our hyperimmune individuals' blood. And compared to normal serum, there's a little to no response. So that's one thing that gave us an indication that we might have something here. And for the somatic hypermutation, Jake?
Sure. With respect to the serum, we chose four species across diverse cobra, coastal type, and crate. Like for neurotoxin example, I think they're about 50% identity at the amino acid level. There's hugely, and yet we saw these extreme potent responses. That doesn't by itself show that they're broadly neutralizing. He could have mounted independent responses, but it does show that he had this immense response, the 1 in 10,000 that was extremely elevated among healthy control serum. In the mutations, using sequencing to look at how much mutation is occurring on average in FVEs versus this hyperimmune subject. Comparing blue to purple, we saw that there was of total somatic hypermutation across the person's repertoire. So in that 16 million or so B cells we obtained when we sequenced them, we see that the mutation in his repertoire panel of tools. The question on antibodies that are against venom, and we tested the biotinol venom from many species and then panning against that total venom fraction to enrich venom-specific binding population. Saw that population was particularly a source of enrichment, a dominant source of enrichment. And when we on the neutralizing antibodies that we're going to share with you today, those were the most mutated of all. And that is he's rotated between the 16 different species over and over again for almost 18 years. And he rotated, if it was an antibody that recognized only a single species, it would only get boosted on that every 16th time. Whereas antibodies that recognized a shared site have a boost, and therefore they would receive more somatic hypermutation attention. So this told us, yeah, we think this is definitely worth building an antibody and then searching through our general strategy. Joel, if you want to go through what our strategy was.
So given that we were talking about the different toxins, we know that this individual was immunized over 700 times. So we would think that his responses were both push towards the conserved sites on all these different toxins, and more likely than not, he had some broad antibody binders here. So our first go through, like Jake was saying, that we were going to go and look at long neurotoxins. Those are the most potent, those are the most lethal. And then build out around that, see what kind of protections that we can find, and see if we can get multiple candidates, multiple antibodies that we can compile into a cocktail that can serve as a universal cocktail against all types of snake venomous bites. So let's.
And the key thing illustrated here also is that if you notice, in our panning, we swap out the ortholog between each round. So we go first against the Cobra version of long toxin, and the second is the third round against the Taipan, the fourth round against the crate. And the ortholog will particularly broadly neutral or broadly reactive binders, and characterize them to see if neutralizing. Is our process. So we obtained 40 mils of blood. That's roughly 16 million repertoire. Sequenced it as we into a library. We knew we had roughly 5 heavy chains and roughly 4 light chains. And so when it's really assembled, 13 phage display, we then transform them into a size of 2e to the 9 to ensure of heavy and light in that combinatorial assembly would be present in the resulting library. That library was then panned on magnetic bead manipulation robots called Kingfisher Flex. The article I described. From that, actually by ELISA for reactivity, and then a subset of those were the sequenced. We sequenced the 3 dominant lineages, and one of those dominant was almost all the 64 active clones. 61 of them belong to this lineage here below. We call lineage D9, and our lead clone FXD9. You can see in the somatic hypermutation. So this clone reminded D9 been subjected to affinity maturation over time, and also was characteristic of a longer than normal CDRH3 with a large consecutive region of non-templated base, which is shown in salmon and is going to get to the crystal. Activity was what the affinity was, these items, and this is really going to into the advantages that and why we built the BLI assay. So our initial affinity studies were done on Viacore. This required that we were able to produce long neurotoxin, so it was in recombinant and pure. To make recombinant neurotoxin, or you need to do a very careful fractionation, or you need to do immunoprecipitation. And in each case, you need to make sure that the material you produced is the pure product in order to get meaningful kinetics. We were very different. So on this table, this is showing the percent identity across the toxins. And these 3 species, the black mamba, Cape cobra, and coastal taipan, they're about 55 to 60 percent identity at the amino acid level. So it's like almost 1 out of every 2 amino acids. That's super different from each other. And yet, that single antibody with 4 picomolar, 7 picomolar, and the worst one was still 490 picomolar against coastal taipan. So binding with immense high affinity against 3 very different, very distant species. So this was actually the affinity molecule that antibodies, like my previous company, we made antibodies for nearly 100 companies, huge antibodies. We've run a filtration campaign, uniformly high affinity molecule that I discovered in my career with this kind of breadth profile. But the challenge of this assay, like I mentioned, it was a significant amount of effort to be able to get kinetics off of each species one by one. And the whole point of this exercise is to be able to survey large venomous species. And so we needed a different type. We're really going to get into the work the assay we developed. So I'll pass through most of this research.
So now comes all the hard, busy work where we need to really scale up our protein production here, because 1, we need to make sure that we can make the same lots so that there's no variation or in between different say, instruments that we're doing to make. So, and not only that, because the Gator uses much less material compared to SPR assays, this provides a really quick QC that we can do, say if a new lot comes out or that we make, and we can easily check it. As you can see here in this BLI graph, we compared against the same 3 toxins that we did during the SPR assays, and it seems to have run at the same, around the same on and off rate, which is actually really good. Although there is no real equivalency between these assays, we can have some kind of comparison, which is helpful. But the great thing about this, and it's useful for me now, is that I can continue to use these quick assays for stability studies, where I keep some aliquots of the older lots, continually check all the newer ones that come off, and we can see how our antibody works over time, in addition to making more for some more interesting experiments that I will continue to.
Yeah, so we use the Gator for, we've been very happy with it. We use it for antigen QC lots, we use it for antibody when we're producing antibodies, and then batch equivalency tests like this, where you're asking the question, is there in some of the proportion activity of which you can set up the ask that question.
Yep. That's all I got for this one, if you have any more. But on to something that I think is a little bit more interesting for everyone here. Now our goal is, do we want to go directly into mice with whole venom? Now, we know we have a potent binder against a long neurotoxin, specifically 4 different recombinant long neurotoxins that are representative and diverse and different enough that we can say that we have breadth. But can we determine that it works against venoms or whole venoms with long neurotoxin subtypes that are maybe slightly different? So here are some of our early studies, especially with the Gator software, of checking against whole venom. So this creates another big problem. We said before that the whole venom itself is a composite of different proteins. And I had my own concerns here that, if we are using a specific probe to load our antibodies onto it, could there be some kind of non-specific binding? Could there be some other things that bind to the antibody in places that we don't want? And going through some of the tech work with Ben and a lot of conversations with our team, we were able to really focus in on some concentrations that gave us an idea of whether or not we can find binding. And so we use this particular assay to help us guide our in vivo experiments. So as you can see through the first 5 or so graphs, that there is substantial binding that we've seen, although we did mess with the concentrations a little bit to really tighten them up. It gave us some really early evidence, or at least encouragement, to go forward with a small scale in vivo study with D9 and a whole venom of a particular species. I will note on the bottom right is the Russell's viper, and it looks really funky for those of you who do a lot of BLI. And for that reason, it's because there is no long neurotoxin. So that serves as a negative control for us and kind of validates how this assay has been working. So.
Wanted to set up an assay. What I wanted was, I said, look, we need to be able to look at least at the 20 species that we know cause morbidity more. I want a screen that lets us look at lots of whole neurobinder, but we're planning to make those 4 others, and we have active programs in line with PLA2 and others. And so I needed an assay to test the whole venom. I needed to be validated, but the figures needed to be publishable. And so we worked with Gator to enable this, and we wrote a piece of internal software to take the outputs, which is sufficient for determining if we're concentrations or affinities or things of that nature in our lab. Looking back, that's fine, but I be nice, and second, it needs to look nice. And so we wrote a software on the Gator. The Gator outputs gets easy to work with because they were on this, so we appreciate that. And these figures represent the outputs of that analysis. What I'm showing you here is the DNA antibody at 2 concentrations that Joel ran here of red and then orange to a negative control, shown in black, against 20 different species of snake. Of the 20 species that we tested, 2 negatives, the Papuan black snake and the Mozambique spitting cobra, that does not have neurotoxin. And then the jury is still out on Mozambique. So we're not certain that that sequence does not, not that it doesn't exist in the genome. The uniprot may not be fully annotated, but it is unclear whether Mozambique spitting cobra, the rest of them, there was this extremely potent activity, and it was consistent with what we saw in the Viacor. Right, I'm sure. Lopids, that's half of all venomous snakes, and you can see we're basically hitting everything except that the 2 non-hitting members in this one kind of outgroup clade, one of which is known to not contain alpha. So this is a standing assay because it enables us to do this kind of high throughput screen on 20 different venoms. In principle, we could do it more. Each of our BNAbs as we move forward. I want to talk about the value work that goes into this type. And partially in with Peter Kwong's group at the NIH. So in order to say, on one hand, we had the comparison to which is great. But on the other hand, I wanted to compare it out to have an independent mechanism to bound the validation of the new assay because we were planning to use this frequently. We compared 2 sets of controls, things that we knew bound, and then 2 sets of things we were marking as not bound. And what we did is we did an immunoprecipitation pull down. The Baoshan at Peter Kwong's group at the NIH did it, and we found for the 4 species that were taken, it's a binder by virtue of the BLI assay. We got a clear for the 2 species neurotoxin. We got no band. So the immunoprecipitation pull down consistent with our assay. Those things that were like the lowest that we considered a positive and the highest we considered a negative, and the assay as expected. Not being satisfied with that, forward and took each one of those bands and sent them out for mass spec sequencing. And by we, again, this was done by Peter Kwong's group, and the results were definitive. Those bands returned back of the long neurotoxin. Is a powerful, multi-validated assay for us to be able to do these high throughput rapid screens for BNAbs that we create and what their breadth characteristic against whole venom to go towards fractionation. It gives us the ability to use that immunoprecipitation assay and it out for sequencing, but also when we have it as a population, we can genetics on that fractionated material. This is important for some of these topics that we manufacture the cells that you're trying to grow them in. Pretty happy about the advantage of the approach is that it tells us we react to something in the venom, the immunoprecipitation, which is more work, which is why it's nice to get that first assay out in the first place to be like, yeah, something in there worth pulling. And for some toxins, logs snake, and so the mass spec will tell, are we hitting all members, and that's a particular interest for us, PLA2, the SVs, and the MPs. So we had all of this. I think what was missing still a question of what is the mechanism of action of this molecule neutralizing, and is it going to be effective in vivo? And that we're going to wrap up and share with you now. Group took this would be sweet to crystallize. No one's crystallized, really reported on a broadly neutralized monoclonal antibody before. And so they crystallized it not once, but 4 times. This is the same antibody. This is our DNA antibody crystallized long neurotoxin, COPA long neurotoxin, MAMA long neurotoxin, and crate mode and all. And if I mode, that's going to reveal to you what the mechanism is of neutralization of this antibody, which is pretty fast. So zooming in here, what you see is that on the left is that's how using crate as an example, that's how it goes and docks to the nicotinic acetylcholine receptor. That super conserved receptor, how it's able to, that's why it stops your lungs and your heart, and you One of these snakes is called a cigarette snake because you have enough cigarette before you die. Like it moves pretty fast, 4 or 8 hours, but it's going to get you. Antibody goes crystals. We can see it binds for all the 4 species that we looked at, and we have crystals. It binds in the exact same mode where it goes into the exact same region, forced to be preserved in order to preserve reactivity to the receptor. Residue on the bottom, you can see by buried surface area, it binds with the proportional weight residues. This thing is acting as a de facto molecular mimic to go block the same conserved epitope of the nicotinic acetylcholine receptor on the toxin. That also immediately implies that these things were, because if it's stuck there, it will bind the nicotinic acetylcholine receptor. And so as soon as we test, we're like, all right, we're definitely going to go try this out in some animals. Copies of this, but it's just the same.
Yep.
So we did this in stages. Let's see, Joel, did you want to go through this one?
Sure.
Okay.
So first thing to do, right, is we made recombinant toxins that we panned against and want to make sure that there's actual lethality with them. So as you can see through the dotted lines here where we had injected the recombinant proteins directly into the mice, these are B6 black mice. They had all induced lethality within 2 hours at the latest. Now, we also want to validate that our antibody can protect these mice. And you can see on the very solid lines at the top that there is protection from lethality with our antibody alone. Now, that seems fun and great, but it wouldn't be as fun if we're not protecting against whole venom. So we move to this next group where we're actually using D9 against the whole venom of the particular subtypes that we made recombinantly. So in the case of Cape Cobra, monocled cobra, black mamba, we see that D9 with those whole venoms is essentially proving life and leaving them alive, which is really, really awesome. I don't know if Jake wants to chime in on any of that.
Yeah, so that basically after testing that, it's able to provide complete protection from in vivo challenge by the alpha toxin against those 4 super diverse snake species that we happen to have the recombinant for. We then went and say, okay, well, how far can this go with protecting? So what we are doing is constructing the molecular basis toxicity. So this is a group with alpha neurotoxin, our antibody against the long neurotoxins alone was sufficient to protect mice from lethal challenge. We've separated, there's going to be other groups we're going to show you, some where there's energy, where there's a second agent that seems to be more. But for 4 species of cobra that span 3 continents, Cape Cobra, monocled cobra, Egyptian cobra, and Indian cobra, we get complete protection, lethality. I think one Egyptian at about a little over 2 hours. Otherwise, they're all protected compared to these mice are all dying slightly different times depending on the venom. It looks like only one mouse managed to escape with its life. The Indian cobra, there was one little getaway guy there, but huge separation. We went out to additional genera. So king cobras are not actually cobras. They're called this, Ohana are called elite cobras. It's a different genus, and we're able to provide complete in vivo protection by Ohana. And also, a third genus. And so this single antibody is able to work against those classes of snake venoms because for them, the predominant source of death, of lethality, is the alpha neurotoxin, the long neurotoxin. And therefore, by blocking it, you are protecting these mice from lethal challenge. That seems to be the drive for those snakes. Now, there's a lot of diversity in snakes, highlighting the regions of the phylogenetic tree that we're protective of the same agent. But, of course, there's diversity in venom. And so what we did is we then went in with our antibody and then a cocktailing it with a single additional thing, or I've heard it called Tirisplata, but I'm not sure which one's right. It inhibits a second dominant venom toxin, to odd manner. It's a molecular mimic of the substrate of that enzyme. And so now with 2 agents, we wanted to ask how much does one or the other or both acting synergistically protect against venom challenge. On the top, you see tiger snakes. see is the dotted red versus the red. There was a little bit of a benefit, but not that much for just protecting from the neurotoxin alone. There was more of a benefit from protecting with virus plotib, although they all die like a day later. Virus plotib is a small molecule, so our reason that let's just do the all-night experiment and keep giving them more virus plotib every 8 hours. We don't with an antibody because the half-life is much better. And then we were able to gain complete protection from challenge, again with agents. A similar story was observed for the inland taipan and the coastal taipan, where a little bit of a benefit from the toxin antibody. The benefit was from virus plotib, and redosing allowed complete protection from death. So we've now extended our protection down to additional gen with consisting of 2 agents. Plotib as a placeholder, we are producing BNAB, the PA2 family as well. Crate, we saw classic synergy where pink line is that the mice, we give them the venom, dead 3 hours. And you give them our neurotoxin antibody, they live 6 hours. If you give them venom and the virus plotib, the venom, they live 7 hours. Either one individually helped, but when you combine our inhibitor with virus plotib, it's a significant extension. Especially if you redose virus plotib. That said, they are still dying, and so this implies to us the third agent that we need to hit. We think it's the toxins that we're not covering yet, and so this is essentially by systematically deconstructing the venom composition. This guides us to a sufficient cocktail against lethal challenge by any snake on the planet. And then these are the tough ones. So these are the yellow sea crate, and then the eastern brown snake, and these show that they're of both ages we have, but the mice are still predominantly dying. We're helping them a little bit, but not much. And so again, we think the cytotoxicity piece here, and that's kind of really fascinating and beautiful that we believe testing this that 3 agents is broadly neutralizing will be sufficiently broad to protect against the entire tree of snakes. And with respect to the Viperidae, it's a similar story. Relate to 2 other agents, but that gets you to a cocktail of 6 and maybe 5 components that could provide kind of global phylogenetic and geographic coverage. What we've achieved so far with NAB and 1 additional agent illustrated here. And Joel, I thought you had some thoughts here, so go ahead.
Yeah, I think the biggest thing here is, as we referenced in the beginning, that this is a tropical neglected disease, and the biggest areas of impact are in Southeast Asia and Sub-Saharan Africa, where there is difficult medical access, not to mention one within 30 minutes. So knowing that we have the breadth between 1 small molecule and 1 antibody to have some protection in these areas is amazing. And it's far and away better than the antivenoms that Jake was referencing from sheep or horses. And we're definitely working towards finding more candidates against all the other different types, because in many cases, if both the PLA2 inhibitor and a long neurotoxin binder do not suffice, the work is definitely not done. But it's definitely a great start. We
think that he definitely has antibodies in the library because Tim lived. So work ongoing. We actually applying our vaccine technology to mice, and that's our day job, is we produce broad spectrum vaccine for influenza, coronavirus, and HIV. Actually very interested in the detecting pseudovirions, and I didn't realize that we can ask like that, but I tempted them, and sometimes but we apply our vaccine technology to mice for broad responses. Mouse antibodies we could humanize back up, but our that Tim probably has the antibodies we need. One of the other characteristics of his antibody and a little bit of our selection process that they're old. So we have 75 Celsius TM and TAG, and we've also done studies that show that we can revert some of what I thought were neutral drift mutations, somatic ARP mutations in the F8. There are some additional antibodies we need to get to go recover those over the next 12 to 18 months to have a product and progressively as we go through the product. Because it's fully human and it's recombinant, we're going to make it full IgG with LALA effector function knockout mutants, and that will avoid a whole bunch of the serum sickness problems And the issue of having to give like 10 vials of antivenom to a single subject, which is typically the case when you're giving Fab2 that has a short half-life. The serum is a big deal, by the way. So if you've just got bit by a snake, so it's worth it, but you're sitting there and you're like, this sucks. And then they're like, oh yeah, no problem, here you go. And they hit you with a whole bunch of horse antibodies. It used to be about 85% of people were having varying degrees of serum sickness to anaphylaxis. It's gotten a little better with the F2 product, and partially getting rid of the FC has avoided some of the reactivity, but you're still being loaded up with like literally a dose of a non-human protein, and that tolerated because the severity of the venom is so severe that you tolerate a whole bunch of side effects of the drug. It's somewhat similar to cancer research. But sometimes people die from the antivenom. There are severe anaphylactic responses. And this is also seen in the veterinary space with dogs. So we think we can avoid that. I went with full IgE because it's human, knocking out effector functions without knocking out the FcRn half-life issue. And that will enable a single dose to provide protection throughout the period of relevant antivenom venom existing, and without the serum sickness. So not just breadth, but also the advantage of recombinant is you can make it a far superior target profile. The last thing I'll mention is that we're aiming to lyophilize. It's really hard to lyophilize effectively when you have polysera. There's resuspension issues. It's not impossible, but it's tough. We have the ability here, these molecules in particular for lyophilization due to enhance lyophilization resuspension with antibodies. And that's important because lyophilization enables you to use dual chamber and to be able to have a dramatically improved shelf life of a product, even outside of reliable cold control systems. And so that's part of the target profile we're going to release. Look, we're not going to displace the current antivenoms now or when we be getting out there, and then we're going to have a manufacturing that produce a monoclonal that meets a 5 million person market. And so I feel that there's going to be and valued use of the existing antivenoms out there. Our objective is to grow up there, and we're doing a lot of work to try to figure out how we can scale monoclonals to serve these dramatically larger communities of need, because I feel that what is useful here at the tip of the antivenom will also be useful in anti-infectives and a number of other areas where biologics could be great medicine. Manufacturer are limiting with respect to the number of doses and the cost of goods to produce them. And that's part of what's exciting about this project. The other part is that antivenom hasn't changed that much in the last 125 years. It's one of the first products that worked. If we can make a work version that's much better, like this could have a very lasting legacy. We could help hundreds of thousands every year for decades, and really centuries potentially. And I think that's a rewarding aspect of the work. But as Joel mentioned, there's work ahead of us. In particular, the lab with the crystallography, the immunoprecipitation bowl, many value strategic discussions. We're putting out a publication now. BNAB, we've already identified against 2 additional toxins. So we're pumping out these broadly neutralizing antibody story to make this kind of a known space realm with respect to these BNABs that hit this classes of toxins across. We'd also like to thank the Bill and Melinda Gates Foundation, US AMRID, Rare, Nimble, and then the founding support from Charles and from Thermo Fisher, who's been some of this work, and Centivax in general. These are all the folks. Do a little special thanks to Mark Ben, who's not here with us, but he was heavily involved, as is John Kim, others that work to enable some of the NIH grant support we've gotten for this and other areas of the effort. And with that, well, thank you all, and we're happy to answer any questions.
All right, I'm back. Thank you very much for that very insightful and detailed presentation. We have a question here to start things off. First off, emphasize cool research! Exclamation point, exclamation point. How challenging are or will be your possible clinical trials in humans? Where do you find these people?
Yeah. So it's just going to control. we're feeling pretty good about that trial because these antibodies literally came from a human already. So we actually have an N of 1 of first in human already established, and we're further making them more safe by removing the effector functions, which is an area where you could run in, and could potentially be an issue. So that's phase 1. What I'm sure you're asking about phase 2 and phase 3, and the where things get interesting, and there's lots of good. So we actually just came back from going to a conference called Venom Week, and then there's a Gordon Research Conference that focuses on venom as well, and had a number of very valuable discussions. Those meetings. The strategy is to have research in India, another one in Asia, one in Africa. There's going to be one in Brazil. We would have sites in the United States, Australia. So those are the major areas. We may do one in the Middle East as well. Where most of your snake bites are rural, and so what typically you have a be near a city, and then within a couple hours of that, you're going to have a series of these clinic facilities, and that's actually where most of your capture incoming cases will be. And so what you do is you set up some of your drug product available out at those sites, and you communication within that network. So as they use it, you would go out and deliver extras. So you have and then your field clinicians are the ones that are intaking subjects. I mean, it would be unethical to try to create a placebo arm, right? That's super. Come in and be like, "Would you be willing to risk it?" So what we would do is we'd have a comparator. So we have the existing antivenom versus our antivenom, and that's typically the arrangement that would be in studies. There are some snakes for which there is no comparator, and so palliative care basically is the for the setup. I think our strategy is to try to go into the large markets where the need exists. Often they do not know the species of snake, so they tend to give them an antivenom if they have it available for whatever it is. So that's the comparator. Our readouts are going to be a little unique because we're addressing not just one little, if you're going, coagulopathy, that's going to be your primary endpoint. It's an easy one to test. In our case, our objective is to be able to treat neurotoxicity or coagulopathy, some other. Create these two endpoints, or point which represents or which is the improvement of it or of coagulopathy, and there's two ways to set that arrangement to be able to get capture that comes in. We're able to affiliate the work with, there's groups now that are creating these little diptychs. They're laminar flow tests, kind of for that information. I clinical study in Africa where the two categories, maybe a cobalt or who, and for the purpose, those are inexpensive. There's companies producing them now, and I think it's to capture that data. And certainly for us to know, is there any snake that, because I think that is just pushed under averaged right now by virtue of poor communication. And I think that frankly is to the benefit of the current anti groups where they're like, the statistical significance, if there's a snake it's not working against, I actually no, that's their attitude. That's the nature of those trials. I will also say that we are in case there's 155,000 dogs in the United States bit every by virus. So those are mostly rat classes. And these anti-venom. There's groups in the United States and in Australia who've been very interested in collaborating with us on testing out our product. Those have some unique manufacturing requirements for us to make it actually feasible in that space. The dogs are either going to have to put up with a human FC. It has the effector functions removed, but it doesn't really have that advanced canonized. We're exploring a manufacturing system which is much cheaper to produce a product which would be appropriate. We're looking now. I idea of going into because we can go in there and it's going to give us a level of quality of data on surpassed by anything we could do with mice or mini pigs or any of the other models, because these dogs are actually out in the field getting bit by a full and variable dose of snakes. And so being able to show efficacy in that space would give me the greatest possible confidence on how we're going to perform humans.
Okay, thanks very much. Another question has come in. You mentioned that you're about to target the other toxin families as well. I was wondering what your approach is for neutralizing the locally acting toxins like cytotoxins or metalloproteases as opposed to the systematically acting long neurotoxins?
Yeah, it's a great question. So I mentioned, so normally, right, with antivenom, what they do is they administer it IV. So it gets everywhere and it gets out to the site somewhat, but you're going to have some enzymes that are acting, they get everywhere. You detect them in the urine, you can detect them in the blood, but there's going to be a local site of those enzymes that are chewing up this. That's why you get a lot of regional tissue damage. I mentioned that lyophilize and use dual chambers. This is still going to subject to us being, we haven't discovered all the antibodies yet. We need to confirm the lyophilize. But the advantage of such a product with a dual chambered syringe with excipient on one side and lyophilized antibody on the other, where it's an EpiPen for antivenom is what it is, right? You crank it over, you shuki it up and stab it in. So it's like tech, very convenient. Ability to administer local to the site so we're currently going to figure that out, right? We are currently assuming some of it goes to the local to the heart from where the bite is, but you could also imagine injecting direct bite, and that would serve to counteract any depot of venom that's just currently leaking out from that site. As we have and serves an in vivo study and probably in pigs where we're evaluate whether the cause problems of is it helpful to go do that? But that is our thinking. Otherwise, we anticipate performing not worse than and potentially much better than the current administered IV, and they kind of get everywhere. It's still the case that our product would enable it for field use. That's both for the people who need this, could be 4 or 5 hours away from, and it's also there's military interest in such because they want to have something where they shouldn't be, or they don't have air support. They administer something. Said the product would also be suitable for IV once you get to a medical center, and of course that would be the preferred when you can, because it gets diffused everywhere and protects possible.
Thanks very much. Next question. Have you tested giving D9 after venom administration? What is the maximum time that the antibody can be given after venom but still retain neutralization?
Sure. So the studies we've done so far are premix. That's the venom toxin. You take the antibody and you administer them IP in the mouse. The study you're referring to is the rescue model, where you give the venom first and then you give the antibody. There are other groups who have done like 10 minutes. They worked with injection, which creates like a slow depot or a sub-Q injection. They could then wait 10 minutes, and I think half of the mice were okay. The mouse is a tough model for us because tiny little guy, so there's actually not that much in space in that model. We know from dogs, from the canine space, and from that when they're bit by a you have a for the word 20 minutes, but most of these snakes you're talking about 45 minutes to a few hours for full paralysis to slowly set in. Often the dogs very often, for instance, they're partially paralyzed. They have a safely ventilate them, and typically the antibodies work very quickly. So the antivenom within 15 minutes they get restoration of ataxia, so they start moving, walking around it. That was to us to see, because I think our antibody likely is like 20 times more high affinity than the antibody in an anti-sera. That just in general, when animals that are immunized, a single-digit nanomolar, they might get down to 500 picomolar, but you don't often see things down to like 30 picomolar. And there's going to be a range in that mixture, where ours is really doesn't have the 70% of antibodies that are against bugs on grass. The immune system of the animal is, and therefore we expect that we would perform not worse and potentially better than those products, and able to reversibly disengage the toxin nerves. And similarly, arguments about that it could damage. There's this whole controversy about this because it does chew up interfaces of neurons and like really any lipid surface. And yet antivenom causes of ataxia, so movement of paralyzed animals, for those bitten by snakes that are predominantly PLA2 active. And that does suggest a reversibility of a lot of the damage that's inherent. That's the window we think we have to really address your question. Yeah, we're going to do the 5 minute and 10 minute on the moving towards the pig model, which is more appropriate for asking those questions of what's your, particularly for really challenging guys.
Next question. Do you think the short neurotoxins are still worth looking at, given neutralization of long neurotoxins shows good protection?
Because these are really detailed questions. Yeah. So here's our thoughts right now. We know just by published LD50s and our own work on LD50, like basically LD50, like MTDs were neurotoxins, simple like in vitro assay that we run. So that's how we tested them initially. Neurotoxins, you'd be potent, and the report is annotated as weak. Them aren't. Also, the short 3 toxins, they actually fall into 2 categories. They're of the same fold, but some of them are actually predominantly cytotoxic as their function, and some of them, we think that's actually why we're not protective against some of those snakes that we showed while we're still deteriorating even when we hit in long neurotoxin, is we think it's those cytotoxins that are causing the damage. We also anticipate, like we go in at 4 times over the LD50 with the mice, but we anticipate as we go to a much higher level of dose, we would expect that the cobras would also show some tissue damage, as is known for clinical bites. And we think the cytotoxins are driving that. With respect to the short neurotoxins, my hope is that they don't matter. I'm wrong about that. And my strategy was, let's go in 4 times over the LD50, so we find the things that you definitely want to block because they're definitely going to kill you. And once you've got those covered, you can increase the dose of the venom. And so this is a systematic deconstruction effort to go, what is the minimum but sufficient number of toxins we know we need to hit? We know we don't, some of them to amphibians, species that are not us. They're pointed at their substrate food sources. LD50s are too far away than what matters for us. There's some of them I don't know. And for the short neurotoxins, I'll say we can make antibodies against them. They're pretty easy for us to make. The other reason we worked on them first was that they were actually kind of a to work with respect to creating forms and tags on them, so we could purify and display them on beads. We can antibodies. Because tail increases the complexity of manufacturing. And so that's why I'm trying to go get the minimum but sufficient set, because there is a cost of good and complexity of manufacture that I'm keeping in mind to create this universal. But you're right. I could be wrong about that.
Couple more quick questions. Easy one, I hope. Do you have a publication on the way with the data?
It's being shopped around just to try to find a good journal to put it in. So that's going on right now. The first is done. The next 2 are in the works. Last
question. Which expression system are you using for your recombinant 3 FTX?
We used 3. They were fine, so we were able to express them just fine. There's some things like PLA2 that does not express well in HEC293. It attacks the cells with vigor. So it depends on which toxin you're working on, how easy it is to perform recombinant engineering of said toxin.
All right, with that, I think, wait a second. Nope, that was it. Last question. And now we're into the conclusion phase. In concluding, I'd like to thank our speakers for providing their experience and insights relating to BLI best practices and how this method can be used in a very challenging antibody discovery program. And I thank you very much for joining the webinar today. An on-demand version will be available in a few days. I'll send a link to it via email to everyone who registered. Please do feel free to watch this version, the on-demand version of this webinar, or any of our other on-demand webinars when it's convenient. Thanks again, and have a great rest of your day.